Thank you very much for having me and giving me the opportunity to share with you a system that I find absolutely fascinating. Now, the disclaimer, which you already heard, is that I have only been working on this since the HFSP project started, which is not a long time. This was very much out of my the systems I had experience with. So I have to say, I know there is a jellyfish expert in the audience, and I will kindly ask her to leave the room right now. Okay, so this is the team. There we go. Okay, this is me from the University of Pennsylvania, and I am a theorist working on physical networks and soft matter. This is my colleague, Annemiek Cornelissen. She is at CNRS, and she is the experimentalist working on jellyfish. And this is Liz Jones for Belgium, KU11, who is an experimentalist with expertise in vascular remodeling and angiogenesis. Now you might ask me, what do arteries and veins have to do with jellyfish? Now, if you remember the horrible title I gave for this talk, you probably know what this is about. If not, you have to listen to the rest of the talk and you will soon figure it out. So this is the star of the show. This is the jellyfish Aurelia aurita, otherwise known as the moon jellyfish. And I want you to notice this beautiful system of canals, the gastrovascular canals. Now, you're seeing the jellyfish now from the bottom. The jellyfish is lying on its top on a Petri dish. Now, another thing I want you to notice here is that it looks nothing like a person, absolutely nothing like a person. In fact, if you look at this tree here, jellyfish belong to a group that could not be further to human than possible. So they belong in a group that includes hydrozoa and anthozoa. And this group has not invented bilateral symmetry, has not invented left-right mirror symmetry like we do. The worm is closer to us than the jellyfish. The frog is closer to us than the jellyfish. The fly is closer to us than the jellyfish. And for this particular reason, I believe that the jellyfish are an excellent system to study fundamental questions about biology, fundamental design principles, how biology solves problems about motion, about circulation, even about behavior. It's also a very, very old group. Jellyfish have existed for a very, very long time. This is a fossil from the Cambrian era, and this is a jellyfish which is about 20 centimeters large. And this jellyfish, to be able to survive, had to have some sort of circulation, some sort of system that will allow it to transport nutrients to its body, because at that large size, diffusion is not enough. Now, despite its importance, not a lot of people work on jellyfish. At least I believe that many more people should be working on it. It was a system that was rather popular maybe in the 1900s, and this beautiful study, the author basically chopped up jellyfish and saw how this would affect how it moved. And also there are some very interesting, very nice papers mid-20th century. However, at the time, people did not have really the tools to study the system quantitatively. Now, more recently, there's been a lot of work on how the jellyfish swims, basically also from the fluid dynamics of the swimming motion and how the muscle that the jellyfish has actuates that motion. But also recently, it has been established, or there is some traction to it getting established as a system for neuroscience, because the jellyfish have an interesting neural net which not a centralized brain like we do, but a diffuse neural net, which the jellyfish uses to control its swimming motion. And of course, here I would have to mention the work of my HFSP collaborator, Remy Cornelissen, who has studied the development of the canals. Now, I also have to tell you a little bit about the life cycle of the jellyfish, because it's very interesting. So the jellyfish begins its life as an egg, basically, and then turns into what is called a planula, which is a ciliated mobile larva. Now, this planula would get attached to a rock and turn into a polyp, which is sessile. Eventually, this polyp will turn into a stack of pancakes. It will strobulate. And these pancakes, which you see here, this is a movie from my colleague's lab, they will detach from this pancake stack and they will start swimming. And they will have reached the motile stage again of the life cycle, which is called the phaera, which will turn to the medusa that we are working with. The sessile and the motile stage of development are so different than people used to think that these are different species. Now, a little bit of an anatomy, and now the actual lesson starts because we're going to be using this later. This is a side view of the jellyfish, and this is the view from the bottom. Now, I'm going to be guiding you through the path of a poor Artemia, which is what the jellyfish eats, through the system of the jellyfish. Now, as the jellyfish swims, it will capture this Artemia, which is little shrimp, and other microorganisms. They will get stuck at these little hairs here, these tentacles, and the subumbrella, the under part of the umbrella, and the oral arms. And at every swimming motion, they will be guided through the oral cavity, through the mouth, which is here, and they're going to pass to the stomachs, which you see here, and through the stomachs, partially digested particles will basically diffuse through the canals, and then they will return back to the mouth. So basically, the easiest way to think about it is the in and out of the jellyfish. The ingestion and the egestion site are basically the same place, which we'll use that later. Now, how does the jellyfish swim? The jellyfish has a muscle, which is lining basically the subumbrella. And you see it here. It's the contraction of that muscle that will actuate the motion. Now, I will show you the jellyfish in action. And this movie is not the best movie you could have, but I made that movie, and I'm proud of this movie, so I'm showing it to you. The little things that you see swimming here are the Artemia. And here you see a very well-fed jellyfish. This is the orange thing. Its stomachs are full. And also, if you wait for a jellyfish to pass in front of the camera, you can also see the canals. OK, so now we became very interested on exactly what the flows in the canals are. This was not the original intention of the HFSP project, but in order to do the HFSP project, we realized that we really need to understand this. And we have been peeling layer after layer after layer since the project started, and we're now much more confused in the beginning. We're doing steps backwards. The only information that exists on this, the only, I would say, semi-quantitative information was from 1955, and this paper doesn't really expect exactly how the measurements were made. There is some description of the flows in the canals and how the circulation happens. It's described that the food comes in the stomachs, and then the food goes through these straight canals, goes distributed in the rim, and then returns through the branched canals. So we needed to understand how this happens. And first, we needed to understand what is the mechanism for flow propulsion. If the so-called boundary conditions, if this was a resistor network, what would be the voltage at the contact points of the resistor network, which is the same place. If that has the same pressure or the same voltage, how does the flow circulate, right? So there are two potential culprits for this. The cilia, and you see here some imaging of the SEM imaging of the cilia from anemic, and also whole body deformations. So which one is it? Now, this question and this dichotomy is not alien to us because we also have cilia in many places in our body, and we also have a heart. We also have flow due to cilia, and we have flow due to essentially a pumping motion. So in the next few minutes, I will be very briefly discussing modeling of the flows due to deformations alone. I will be ignoring cilia. I'm allowed to do this because I'm a theorist. So first, as you saw in the movie, you can believe me that there is very large body deformations as the creature is swimming. And the canals change radius. To understand how this, what happens, first we need a model at the single tube level. If the canals change radius, we know how to do it. In fact, people have been doing it in the literature since a very long time ago, 1967, et cetera, et cetera. This is called radius-imposed peristalsis. The canal changes radius, there is a passing wave, and it pushes the fluid. The problem with the jellyfish is also the canals are changing lengths, and for that there is almost nothing, basically nothing in the literature, and we had to do the math. For the ones of you that like these sorts of things, for the radius-imposed peristalsis, you have mass continuity, you have equations for the momentum, you have equations for the flow force balance, which includes fluid pressure, balancing external radial force per area, the squeezing, and then elastic stretching form. But the problem is, if you have length-imposed peristalsis, you really have to worry about the movement, the longitudinal movement of the wall. And if you have a no-slip boundary condition, then it becomes a very hard problem. And you have to be very careful with regards to Lagrangian versus Eulerian description. Anyway, my student, who is amazing, figured out how to understand this. Now, we have to take this single pipe and make it a coarse-grained model, combine it to understand the whole network. We have to put many of these pipes together and model the whole contraction. And this is the step that we're currently doing with data. We have to actually feed in this model the lengths of the vessels over time, the lengths and the radii, from the deformation information of the actual jellyfish. So this is done, is being done right now. This step 2 is done. So let me discuss a little bit the physics of the basic principles for deformation-induced flows. So this is the jellyfish. So every time the jellyfish swims, some fluid is coming in at part of the cycle, and some fluid is coming out. Now, if the jellyfish doesn't have some, at least small, net unidirectional flow in the canals, what's going to be happening is the jellyfish is going to be taking a shallow breath, in quotes, and then a shallow breath out, and the fluid and the food near the ring of the jellyfish would not get anything, and the jellyfish would die. This is not an efficient way to do this. You need somehow, by you, I mean the jellyfish, to establish some sort of net unidirectional motion, like the one that the paper from 1955 had found. Now, I'm a physicist, and I can do the spherical cow of the jellyfish, which is a radially symmetric jellyfish. Now, if I have a radially symmetric jellyfish and a radially symmetric motion, as we saw, you can't have this net motion. Nothing breaks the symmetry. So the jellyfish would starve and die. But if you look carefully and not be a physicist, but be a real person, we see that the symmetry here is broken. There are two kinds of canals. There are the straight and the branched. And in fact, I can make a simple model here, and I can say this node here, which is the node that goes to the mouth and to the seawater, is this one. This node number 4 is this one. Node number 2 is this one at the radius, and another node number 3 here is the one also at the radial canals. And I can make this link here thicker, more higher conductance, because there are more vessels in these branched canals. And then I can make a minimal model of the circulation of the jellyfish, which is 1D, and you can look, if you watch carefully, the phase difference between how things elongate and how the things shrink. Now, this is one way to do things. Another way to do things is not to worry about breaking the geometric symmetry, but directly have a peristaltic wave that goes around like this. This one breaks the symmetry and has a hope of doing efficient circulation, of making a jellyfish that can feed itself. So let's try here and break the spatial symmetry. So I've broken the spatial symmetry. And you see this jellyfish here, this computational jellyfish is swimming like this. Now this jellyfish, though, has a problem. If I were to play the movie backwards, you wouldn't be able to tell. There is path reversal here. And I forgot to tell you something very important. The flows in the vessels are Stokesian. They're very low Reynolds number, meaning inertia doesn't matter. So for these flows, you might have heard the so-called scallop theorem, that when the flows are very viscous, if you have a path reversible swimming motion, you cannot swim, right? If I were to try to swim like that, or like that, I wouldn't be able to do it when the viscosity is high. So for the jellyfish, it's like we can rename this to the umbrella theorem. So when the jellyfish is contracting in a path reversible motion, the jellyfish cannot have net flow in it, because whatever happens in half the cycle will be undone in the other half. And we can see this if we look at these nodes here. If I am to integrate the output of the straight canals and the output of the branched canals at the site that contacts the seawater, I would get net zero. Nothing would happen for its case. So this jellyfish is bound to die. Now, I can design a swimming motion that is not path reversible. And in this case, I can play this game, and I can see that there is a little bit of fluid flow that will go through the system, and at every iteration, it will move, and 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 it will find its way in the system. New food will reach the end of the jellyfish, the rim of the jellyfish, and come back. So it is possible for a jellyfish to feed itself only with deformations. Now, of course, there is something else. This is the other mode I warned you about. And this is this swimming motion. This is from a paper at PNAS, and those jellyfish use this type of swirling motion to navigate around obstacles. Unfortunately, the ones in the lab of my colleague only do this when they're very sick and they're about to die. But according to our theory, this type of peristaltic motion will produce net flows. But it's complicated. OK, so now we need a reality check. And we need data. So I will be showing you some very, very preliminary data. This here is the anesthetized Aurelia. The movie is playing. So this is a jellyfish that's lying on a Petri dish. You see it here. This is the microscope up there. And if you really watch it carefully, you'll see that it's anesthetized, but it's moving a little bit. Here I overlaid 1 second after in blue. You see there is really not much motion. And here in this movie, you have to squint and look here. These are food particles that actually do crazy things in the anesthetized jellyfish. This particle here moves a little bit up, then it will do a U-turn, start returning for no reason whatsoever. And here, if you look carefully, you'll see the same vessel, particles going simultaneously one way and the other way. It's like there are lanes inside the channel, which is something we don't understand at all, and we're trying to get measurements, get resolution in the depth of the canal and see what's happening. This is here, the jellyfish near the rim, at the end of the, the furthest away from the mouth. And you see these rotational flows. I'll play it once more. Here, these little vortices, but basically the fluid is not going anywhere. So this jellyfish doesn't follow what was in this paper. This jellyfish would not get food, and this jellyfish would die. Now, this is the awake Aurelia. This is the non-anesthetized. Here, you see some of the Artemia that got also, as the jellyfish was scooped out of the tank, they also arrived with it. But you see how strong the contractions are. And here is a close-up. And I want you to notice two things. I want you to notice how much the vessel is changing, the radius of the vessel is changing, how abrupt the motion is, but also the unidirectionality of the particles. It's difficult to see it, actually. And the fact that it's difficult to see it makes tracking these particles very difficult. You need to do image stabilization, and you need to track the same particle during these violent contractions, and this is something we are working on at the moment, but there are a lot of technical difficulties, obviously, because this is, again, this is moving a lot. However, from the few particles that we have measured so far, we see that the average particle velocity, the typical particle velocity of the anesthetized, in the anesthetized jellyfish is 50 microns per second. In the awake is 4 times higher, approximately, which is very interesting. Now, what is also interesting is that the non-anesthetized jellyfish can maintain this velocity also in between contractions. Something really interesting is happening fluidically that we're trying to understand here. So now, there are 2 things that that were the main message of my talk. The jellyfish needs to swim to produce an effective circulation. A jellyfish that doesn't swim will die. And not all swimming motion, though, is created equal. Only the ones with path asymmetry can produce an effective circulation. Not all swimming motions will do. Now, where we're going, and I'm going to do my last few minutes, I will do a really quick tour and try to put this in a broader picture. I've talked to you about swimming jellyfish. I've talked to you about cilia. I've talked to you about hearts, which is something that my group was very interested in before I got into jellyfish. So biology needs flows to survive. If you're larger than a certain length scale, you really do flows. You really need flows, and you need to power those flows. And there are many different mechanisms to power those flows. There are whole body deformation, cilia, hearts, multiple contracting vessels like the earthworm, peristalsis with valves, which is also something that we have worked on in my lab, which is in our venous system and actually the lymphatic system. And I think we're going to hear something very, very interesting about the lymphatic system later on, which does not have valves. We're going to also, another system that our body needs to power is in the gut and in the esophagus, where we have large amplitude peristalsis. We heard in this meeting about shuttle streaming, also motor. So there's all of this array of ways to power flow, and it will be very interesting to understand when do you use one versus the other and what are the unifying principles. Now, where it all started also, we met at a conference. It was right after the pandemic. October 2021, we were still wearing masks. It was a summer school on hemophysics, and I was very interested in this problem, in the problem of how dynamics interplays with development for flow networks, and how does this affect the problem of vascular shunting. So we're doing theory on this. I met Liz at that meeting, who was interested in angiogenesis, and I started talking with her. I told her, can you do experiments where, with patients with LVAD, left ventricular assist devices, where, if you have access to that pool, maybe blah, blah, blah. So we started establishing a discussion, and then we saw, and Amit had a poster who was talking about the jellyfish, and then we realized perhaps the jellyfish has to solve similar problems like us. It has to power flow with a pulsatile motion. And then we were trying for a long time to find a vehicle, something that would fund an international project, especially when one of the partners, well, actually two of the partners had never worked on anything related to jellyfish, and we had no preliminary data, and this was our solution. So we're very, very grateful. Now with that, I will finish because I know you are all very tired, and I would like to thank the groups that produced, the members of our groups that produced this work. Since I didn't show any of Liz's work, it's not here, but from my group, it was primarily Aaron Wynn and Paheli Desai Chowdhury, and this is people that contributed to the experimental aspect from Anemic's side, and thank you very much for your attention.
Thank you, Eleni. Fantastic talk. Dare I say, because of just the fact that you started only last year, I mean, this was really awesome. Questions? We have time for 2 quick questions. Okay, go.
Beautiful work. What's the Reynolds number associated?
Can you speak up a little bit?
What is the Reynolds number associated with these jellyfish?
That's a very good question.
Microphone.
It's below 1. I can tell you exactly, but it's below 1. It's very low.
Yeah, and also just a quick question: is the circulation, the frequency of circulation, the peristaltic motion, is it resonating with the full body, or is there some parametric resonance that happens in the
That's a fascinating question. I don't know, but it changes with age. So I don't know, but something very interesting might be happening because it's different in small jellyfish, and it's very different in large jellyfish. And you can see it in the movies, which were all real time. Very good question.
Last question before the coffee. Very nice talk. I was wondering if you know how this peristalsis is initiated, if it's like in mammals, it's neuromuscular thing, and if there is any pattern also in this situation. We are not working on this, but I know there is at least one group that is working on this at the moment. The difficulty that we have, and many groups that work with this have, is you cannot study this in its natural environment. It needs to have this particular motion to swim, but then you cannot record. We cannot record the flows in the freely swimming jellyfish, and I don't think you can record. You can do calcium recordings in the freely swimming. But people record the motion and they record calcium, and they are trying to make maps between the two. But there is some work, and if you want, I can give you some papers. But this is not our group.
Okay, thank you. So let's thank all the speakers of this afternoon session. Wonderful.